I'm Dr. Jamie Foster. I'm a forage agronomist with Texas A&M Agrilife Research at the Beeville Station. Hi everyone, thank you for being here this afternoon. It was quite an honor to be invited to come back to my alma mater. I have spent some time as I prepared my presentation today reminiscing about my time here. And I wanted to share a little bit about that time before I share the science with you all. So my plan today is to talk to you about my time here at Tarleton and how it really shaped where I'm at today. And to share with you a little bit about the Beeville Station. We're a large station, but we have very few people. So I just want to let you know what we do there. And then share with you a little bit about some of the research that I've done in the last 15 years that I've been at Beeville Station, and one big project that we'll be taking into the future. So it felt so good today to walk out of this building and look across to Humanities and see the library on the right and the brick on the walkway. It was a little bit of a deja vu feeling because I hadn't seen that view so many times before, but it's been a really long time. I graduated from here with my bachelor's degree in 2002. So it's been quite some time. These first few slides are a little wordy. I'm a little nervous sharing about my personal life, probably more so than I am sharing with you about science. So I don't want to forget anything. But I was a first generation college student. Anybody here in the audience also first generation? Okay, so you understand the challenges that come with that. Having to figure out the form or who to call or where to go and how to get information, because sometimes when your parents have been through the system, they have that knowledge, they can help you out a little bit. But you're kind of on your own when you're first generation. It seems like your family supports you and they are so proud of you, but they don't really get it. But I enrolled here my sophomore year. I had earned enough money from my sheep sale at San Antonio Livestock Show to pay cash for Austin Community College. I lived north of Austin at the time. But I really loved my time in FFA when I was in high school, and I thought, I'm going to be an ag teacher. So I came here, I signed up to be in ag education, and I found out I was going to take welding, y'all. Welding in high school went very horribly badly. And so the only option I saw was to change my major. So I changed to animal science within the first month of enrolling here. And also within that first month, my dorm roommate, who I'd been matched with at Crockett Hall, whew, it was bad. And it's been razzed, and you have some great new buildings across campus there. But she said, oh, let's go check out Sigma Alpha. I know there's one sister here in the audience. Any other Sigma Alpha sisters here? All right, so it's Sisters in Agriculture. It was a great group for philanthropy and support. And so we went, and I found this. I dug these pictures out from being a duck camp leader in Sigma Alpha in my purple book from 1999. I still have it. It's in a box. Anyway. Okay, so we joined Sigma Alpha, and I met a friend in Sigma Alpha, and she worked in Dr. Jim Muir's lab at Texas Agriculture Experiment Station, which is now known as Texas A&M Agrilife Research and Extension Center at Stephenville. And so she knew I needed a job because I had changed my major. It was going to take me a little more time to get through school. And so she talked to Dr. Muir, and he said, sure, bring her over for an interview. And I got the job. So she and I and all the other grad students that we worked for did some really crazy things. We went out and collected wild legumes in bar ditches. We had to collect burr medic seed, and then we had to break it open and count the seeds inside of it. So we made the best invention ever. And we took our socks off and we padded the medics so they would stick there instead of picking them individually. So you can get very creative when you're desperate. We carried around a big, huge scale on wheels around a pasture to round up goats to get their weights. Built a lot of fence. We had a good tan that summer. But we loved it. We loved the science part, the manual labor. And We also had hands-on opportunities in classes. That was one of my favorite things about Tarleton. So I took beef cattle production, and I got to go halter break a heifer and draw blood and all sorts of other things. Dairy science, we had to be at the dairy at 5 a.m. to milk cows. So those sorts of things were really important, I think, if you're going to get a practical education in agriculture. So we enjoyed all the traditions while we were here, things you guys still have the opportunity to participate in here. Beat the drum, cruise the island, which I guess it's different now than it was then. Making poofs for our homecoming float, being a duck camp leader, those sorts of things. And then graduation day came. I had put in applications with NRCS and a few feed companies, but I hadn't really had any hits there. And towards the end of my time as a Bachelor of Science student, Dr. Muir called me in his office and let me know about this program called Pathways. And this still exists. If you get your bachelor's degree from a system university, you have the opportunity to go to get your master's degree at Texas A&M and College Station, and it helps pay for part of your way there. And so I had worked for the graduate students, chasing goats and putting out hominy feed and grinding their samples, but I never thought like I could be one of them. And I was really taken aback that he had asked me if I might want to be a grad student. It just sounded like something for, I don't know, other people. And so I thought, well, this sounds great. So I did my research here, still in Stephenville, so I got to be back and forth. And then I presented some research at the International Herbivore Symposium in Mexico. And before that, I had traveled outside of Texas twice in my entire life. Once to Louisiana when I first graduated high school, and then another trip for Sigma Alpha National Convention while I was here, because there was funds to pay for that. But while I was there, I met Dr. Adesogan with the University of Florida, and I really liked him and his research focus, and I asked him, well, are there any opportunities for a PhD? And he said, if you can pay your way. So I applied for a fellowship that I then got, and I was able to continue on for my PhD work. And through my time here at Tarleton and with Sigma Alpha, I really got out of those experiences what I had put into them. So I really just want to stress from my story is it doesn't matter if you're a first generation college student, you could be a sixth generation Texan and a fourth, state of Texas Texan and a fourth generation Tarleton State Texan, right? You still have to put into something if you're going to get anything back. Really take the opportunities that you might have here because this is a fantastic place. So I feel like my time here really changed the trajectory of my life. I would never have a PhD if it weren't for my opportunities to have met my sorority sister and get a job at the research station and use my socks to harvest medic seed, those sorts of things. But so that was my story. Thank you all for listening. So now I'll move on to sharing with you about Beeville Station. So who knows where Beeville is? I know that one person in the audience is from there, so they know very well. Okay. So I like to say that it's about halfway between San Antonio and Corpus Christi in South Texas. So we are in the Gulf Coast Plains region. We have heavy clay soil with high pH. And here's a map of our station from Google Maps. So we are, and anybody who's ever been to the station has heard this, but we're actually the oldest agriculture experiment station west of the Mississippi River. Our gate opened in 1894. It opened as 151 plus a little bit partial fraction of an acre. And they did a lot of vegetable and fruit production research. In the 1960s, some of the notes from the very first scientists were retyped for record keeping. And you can read about spinach and peaches and all sorts of things that we don't grow there anymore. But in 1980s, the shift to forage and cattle research occurred, and there were two primary scientists with 100% research appointment that worked there, and it's been that way ever since. and we were gifted land. And so our total acreage now of this farm is 1,322 acres. And so I manage that. I've been the station director since 2021, so it's been 3 years now, when there was a retirement from my counterpart in animal science. And so we've recently done a strategic plan, and we really want to use this land to promote natural resources management for the coastal plains with a focus on animal protein production. So that linkage between forage agronomy and animal science is very important on our land. And we would really like to translate animal plant soil nexus research, because that's a very unique system to look at all three components, to both students and through extension. In our strategic planning, this is kind of what we developed, but I just want to really stress that what we are building at this point is animal plant soil long-term systems research. So these are the people that work with and for me. We have Hamad Saeed. He's a research scientist. My counterpart in animal science was Dr. Milton Thomas. He passed away this February. And Dr. Thomas had hired Dr. Saeed, and he's a data scientist with a specialty in breeding and genetics. And so he's there kind of helping me manage the animal science component while we have an open position at the farm. And Carlos Serna is our farm foreman. He does our cattle checks every day, make sure everybody's fed and happy. And then Jason Vasquez, he's our agriculture research worker. And then for my forage program, I have a long-term partner in crime. Her name is Brittany Garza, and she's the senior research associate for the forage management program. She is actually a graduate of Texas A&M College Station, and she and both of her sisters all have worked for me through the years. They're all great, hardworking individuals, but I'm working with several graduate students. I have 2 listed here that do their work at Beeville or on projects that I have at Corpus Christi, but I'm on committees for 12 or 13 graduate students. I've always got several undergraduates helping at the station. So, I cut that T off there. But, and this summarizes the 3 main components that I have within the forage management program. Where we're at is a very diverse area of Texas. We have row crop land that's primarily cotton and sorghum production. And then we also have introduced pastures and native rangelands. And so as a forage agronomist, I'm trying to meet the needs of a lot of different stakeholders and to solve problems. So it can be a little diverse what I'm actually doing on a daily basis. But integrating legumes or co-products, I'll share a little bit about that today. Forage evaluation of native and introduced species for livestock, wildlife, bioenergy, bioproducts, cover crops, or other uses. In emerging technologies, I'll focus more on this topic area today with remote sensing or other opportunities for forage management. And so the title of my talk is that forages are a keystone component of the grassland ecosystem. So this is a graph of an arch with a keystone, and that's forages. At the base of each end of our arch, soils and livestock. So again, remember plant, animal, soil nexus. So forage and livestock interact with all these ways that we manage grazing, manage the land, the quantity of forage, canopy cover, nutritive value, stocking method management. Soil and forage interact with the primary input, being the forage into the system, how that interacts with nutrient cycling, water infiltration, etc. And these all work together, kind of influencing each other in all different directions. And so this summarizes it. I'm sure in a class you have seen this graph before, right? So I just want to remind you of it. So when we look at a grassland livestock, we could add soil to that, to the ecosystem, then we know we're really looking at energy transfer. And what is our energy? Carbon, right? And so we have to have our primary producers, all our plants, forages, to feed our primary consumers, and then the secondary consumers and the tertiary consumers. So forages are fundamental. So when we look at carbon cycling within the grassland, we know that about 10% of the global soil organic carbon stock is held here and fluctuates there. Our sources are from respiration, eruption, and flatulence and excrement, soil and forage respiration, fertilizer equipment, forage decomposition and burning, and our sink or our holding of the carbon is in our plants as they photosynthesize, because again, the primary producer, the soil, nitrogen fixation, and organic fertilizer amendments. And again, I'm sure you guys have seen that in class pretty recently, but just a reminder. So I'm going to talk a little about some of our work at the forage and soil nexus, and then I'll talk about some forage animal nexus, and then we'll bring it all together. So soil organic carbon in recent years has become something everybody is talking about. And the reason that it's important is because it's the basis of a lot of the ecosystem services that our soil provides. So you cannot have a low soil organic matter soil that is holding on to water, right? Because soil organic matter, the soil organic carbon, is where the water is held in our soil. You cannot have sufficient microbial activity in a low organic matter soil, right? Because that's their food, the energy transfer that's happening in the system. And so this just summarizes what's happening to the chemical, physical, and biological components of the soil when you have organic matter. Chemically, you have enhanced pH buffering, enhanced nutrient cycling. Physically, you have a better structural stability of the soil. You get a sharpshooter or shovel out and you dig your soil and it's going to stick together as aggregates. It influences the water retention, so the water that can seep into the soil and then the water that's held. And biologically, back to our microbial function and nutrient cycling. So carbon credits are becoming something very important for grassland managers. So this slide really just summarizes that if you have a grassland that you're managing well, that's in a perennial forage, you've probably already got a high soil organic matter concentration. And so some of these programs are targeted to increasing the soil organic matter, but you can't do that if you're already kind of at your capacity. And so if you have a row crop land, and I'll speak about this in the next few slides, but if you have a row crop land where you're doing full tillage, your soil organic matter might be here. And you convert to a perennial pasture system and give it some time, and your soil organic matter will be here. And so you can easily incentivize that by measuring carbon in the soil and then going back after a certain amount of time and measuring it again. But in a grassland, how do carbon credits really benefit our landowners if they're here and in 99 years they're still going to be here? And so that's an issue that we're working on as a team with collaborators Katie Lewis, Joseph Burke, Paul DeLon, Jordan Bell, and Srini L. So they have gone out and measured soil organic carbon to a 90 centimeter depth all over Texas. And this is supported by 3 commodity boards. So this is a great example of bringing different things together. but also they're measuring grasslands, because the thought is that these perennial grasslands might be examples of where we cap out our carbon. But there's not a lot of data. So I found very few papers on rangeland soil organic carbon concentrations, and even fewer on introduced grasses, introduced grasslands. So what we know is that our cropping systems have a greater potential to store atmospheric carbon than the values reported in modeled systems. And this is some of our data set from the first year. But the total acres that are represented by the sampling efforts is 18 million. So it's quite the effort. And that's based on the region and the soil series type, and then how many acres of that is in Texas. So what we found when you look at the South Texas data in the Gulf Coast, we look at corn, cotton, introduced pasture on a Parita sandy clay loam, and that's at Beeville Station, and then corn, cotton, etc. But the perennial pasture has twice the organic carbon than an annual row crop. It's not really a surprise, but we have a value to that now, that in that year and that climate at that time, this is what we have. So that's great. So why does that matter? Again, the physical, chemical, and biological things that are influenced by the soil organic carbon, like water, right? And so what we know is if you have 100 pounds of soil, 1 to 2% of organic matter can hold about 35 to 45 pounds of water. But you double that, and it's 165 to 195 pounds of water. That's a lot more water available for your crop production. When I say crop production, I don't just mean corn, or sorghum, or cotton. I mean your forage, your food for your cattle, your beautiful carbon input into your grassland ecosystem, right? So in addition to working in forages, I also work a little bit in this tillage trial. The reason I got kind of stuck in this tillage work is because what I cared about was trying to add in a cover crop, because the cover crop was a forage. And then there might be potential to build some fence and put in a waterer and bring in animals and integrate row crops and livestock. But my cover crop in this environment was a miserable failure. But I was so interested in the soil plant component that we just continued working on this. So in 2008, which I wasn't hired yet, I was hired in 2009, this study was started as a demonstration site. And then I had a graduate student hired in 2011, and that's when my work began on this. So it's a pretty long-term project. It took 10 years of soil sampling to prove that we could increase organic matter if the only thing we changed was our tillage, from full tillage to no tillage. And so we were wondering, well, can we do this faster if we add in more primary production of carbon to the system? So we have some work on that too. But simply changing your tillage practice was a good economic thing for the system in South Texas. Mac Young, he is since retired, but he wrote a nice paper in Farm Assistance Journal that switching to no-till cultivation could maintain or improve the yields, reduce production costs, and increase the profitability per acre. And the reason for that is primarily because if you're not tilling, you're spending a lot less fuel, a lot less time, on your farm. And there's a great program through the field print calculator. The blue area here in the middle of my spidergram, and I have things like land use, soil conservation, soil carbon, condition index, energy use, greenhouse gas, water quality, and biodiversity. And these are all based on NRCS and other metrics. You can input your production data, and this is your output. So what we know is that we have a more we have a smaller box here, which is a reduced footprint and a better conservation score when we reduce our tillage. That's really no surprise. And these are based on 5-year averages from the USDA Ag Statistical Service. So remote sensing, we'll get in more to this. So we flew this project with a drone, and this was the output here. So when I go to the field, we plant in February, we get our sorghum out in July, and our cotton out in August. It's hot. But you can't skip. So in my plot, it's about 120 feet long, 16 rows wide, at 3 feet, about 38 inches actually row spacing. I might take 3 to 4 samples to know what my yields were. I'll soil sample 3 to 4 locations within that area. So when you fly the drone, you can break that out into 1 by 1 meter grids. And so it took 10 years of collecting data to show that these were different biologically, right? And then this wonderful student, Dr. Akash, because he received his PhD, proved it in 1 summer because the visual indices that came from the remote sensing images proved that there were treatment differences that we could not measure with our traditional methods. So we had another study at Beeville, also cotton sorghum rotation, where we included different legumes and strip tillage. Again, we're trying to add in more primary input to the system. This one really didn't change our soil organic carbon over 8 years. That was as long as we had funding for this project. But what we did prove is that it didn't reduce our yield. So that's something that a lot of producers are worried about, rightfully so. Are cover crops going to decrease my soil moisture or impact my yields? And what we did find, though, interestingly, with these legumes being added is that we had a kind of a binding of our nitrate nitrogen. And so what we need to be really conscious of in these systems is that we are fertilizing at the right time. So we're not getting an imbalance in our nitrogen and our carbon. And again, this system, when we reduce tillage to strip tillage and we add in our legume cover crop, we also improve our conservation score for the system. So I'm leading the grasslands commodity team for the Texas Climate Smart Initiative. This is statewide, providing incentives to landowners. And what this does for me as a scientist is provide a database. It's all unlabeled. All we're going to know is county and soil type about the carbon. So we can get some better comparisons of what's going on in our grasslands. Because obviously we know management is impacting that. So that's a little bit of my work in the row crops and grasslands. So forage and livestock. My master's degree work and some of my PhD and postdoc work was in situ rumen degradation kinetics. So what's happening in the rumen when we put a bag with a forage sample or feed sample inside of it. And that's kind of my expertise there. But I'm not sharing that work today with you all. This is more of a indirect measures of what might be happening with our animals. So Old World blue stem. Is anybody here familiar with Old World blue stem as an invasive weed here in Texas, Australia? Dr. Muir, of course you are aware. So in South Texas, this is really bad. It's really taken over a lot of acres, but nobody can tell you how many acres, right? But it was seeded into right-of-ways and it marched into pasture lands. So they're not natives. We have wonderful blue stem grasses, right? Our big four contain 2 blue stem grasses, little and big blue stem. These aren't native. They're introduced species, and they're more than one genus species, more than one ecotype. And they're naturalized. So several places, USA, Australia, Mexico, Central America, and West Indies. So we're not the only ones in Texas facing the issue. So the reason it's a problem, 2 reasons it's a big problem. It becomes a dense monoculture, and I know there's a lot of wildlife students here, and so you've learned that dense monocultures are not good for the system. It's pushing out things like quail. Like at Beeville Station 15 years ago when I was hired, we heard quail a lot. Now it's pretty rare to hear them because we have had old world bluestem march through the place. But the other thing is, I think it's good to balance wildlife and livestock, but if you're concerned only about the livestock component, it has a very low nutritive value. There's times of the year, and even some of the hay we end up putting up, our crude protein is 4%. That's below the minimum required by a mama cow, right, to support herself and any physiological stage that she's at. So even if she's dry, not pregnant, it's not enough to support her. And so in a warm season grass pasture, well stocked, you'll see about 2 pounds a day gain. It's almost half that on an old world bluestem pasture. So, and it doesn't matter if you reduce your stocking rate, because then the seed, it just goes to seed, and the nutritive value lowers even more. So there's 2 types that we're tending to fight in South Texas, Bothricola and Dicanthium. We say interchangeably yellows or KR, and then Clayburghs. So there's a kind of a debate on whether you can tell these apart. I cannot, but I have a colleague, Dr. Megan Clayton, and she can. So we have the KR or yellow bluestem at Beeville Station. So they tolerate dense soils. They're very grazing tolerant. They withstand close grazing. They have phenotypic plasticity. So if you mow or graze it, instead of growing upright, it grows along the soil surface to hide itself from further mowing or grazing. You get a rain, and it's like you blink, and it's got a seed head up. It's producing a lot of seed. So what we know is the qualities of good forages. The reason it was brought here is because it's the same as the quality of a good weed, right? Unfortunately. So they're not palatable. They lead to selective grazing, which then further perpetuates the problem. You boot out some of the wildlife. Some research in other states further north implies that if you can keep it grazed to 4-inch stubble height, that's better. It's really hard to do that. You have to have a lot of animals to maintain it. And then when it gets that height, what do you do with the excess animals? So it's very difficult. And the reason that recommendation is made us here is that if it's maintained low and repeatedly grazed, the nutritive value tends to be a little better. So all that said, we did a project that was funded by Texas Parks and Wildlife where we did a shotgun approach. So we had plots where we combined treatments over time. We burned it in the summer because there had been some research that said that that might be helpful. Then we applied 2 different types of herbicide. We tilled it, we mowed it, all these things kind of overlaid on top of each other. You know what we found? It almost brought us to tears. So after 18 months, it was back at a higher canopy cover percentage than it was when we even started. Okay, so I don't have any good recommendations to make to get rid of it. But what we can try to do maybe is learn more about it and manage it, work with it. I don't know, we'll see. So I had a graduate student, Dr. Shin is her name now because she finished her PhD work and this was part of it, and she moved back to China. But we wanted to know, have this area where I had planted Tifton 85 Bermuda grass in 2012, and the older bluestem had kind of started to encroach from the front fence that went right through this pasture area from the roadway. Could we identify what was happening there? Could we see some more information about the spatial distribution of the spread, the impact of management, strategies to work with old world blue stem? And so one of the great things about being there in South Texas is that we have two great universities there, Texas A&M Kingsville, where they have a good College of Agriculture, and Texas A&M Corpus Christi, where they have really good engineering program. And you've probably seen them, the Hart Institute on Shark Week too, from Texas A&M Corpus Christi. But scientists from both universities and at the Texas A&M Agrilife Research and Education Center at Corpus Christi, which is our mothership for Beeville Station, got together and began a program with remote sensing. So we have people like me, biologists, that can help understand the data that they're getting. But also we have engineers who use cool 3D printers to make the right prop to get the drone to carry the payload we need it to carry. Or we have a data scientist who can build an awesome graph to show a heat map of what's going on. So it's a big team effort. I just want to share that. But Sylvia did build the algorithms to translate this data. She built a workflow to differentiate between grasses. So here is, she used two different types of machine learning methods, support vector machine, and this was August, and then we go along through the year, and we end at the very end of November. So our first frost date down south where I'm at is right at the tail end of November, about November 30th, okay? So that's why we ended the trial then. And you can see the differences that's going on in the field. Green is Bermuda grass, blue is our yellow blue stem, yellow is Johnson grass, red is ground, and then we have our plot boundaries here. So we went from May through to November. And then she compared that to what's called a random forest. It's through the same time period. And she found accuracies are listed here, overall accuracies. So the ideal date to tell yellow or KR blue stem from Bermuda grass or Johnson grass is around October. So this is the red, green, blue image. So it's just like taking a picture with your phone, or if you still have one, your camera. And the reason that October is a good time to tell the species of grasses apart is because at this date, the old world blue stem had gotten this characteristic red color. You can see it in this picture. And when we began the remote sensing program with forages, I had met with a scientist from the Midwest, Kevin Price, and he shared with me that if you can see it in a picture, you can also develop an algorithm to draw out data from the image. And I think that was pretty profound thought. So you can see the differentiation here. So our accuracy around that time was in the mid-80s. That's pretty good. If we were able to translate this up to satellite level, that would be really cool. It would be helpful to have two pieces of information. It's a regional issue, so there's not a lot of grant support for it, but we know it's impacting a lot of people. If we had information on the acreage and the economic impact, right, then we could prove we need support to solve this problem. Okay, so I'm going to shift gears to the forage-animal-soil nexus. And it's going to tie in what I just shared with you, the remote sensing, to the future work. And so this was our first calf that was born this year, calf 001. So the animal scientist who passed away in February planned for fall breeding, and that calf was born just over a week ago. And here I am, we're just running off to Stephenville, leaving everybody there to deal with the new babies. But he designed this important research, I think, for the future of Beeville Station. And Dr. Hamad Said is continuing it on. We're working together, and we hope to hire another animal scientist that will help continue this. But we have mixed breed cows, so it's a motley crew that we have now. But they're all very important breeds for heat tolerance. And if you've heard of the Ag Research Service Meat Animal Research Center, the U.S. MARC program in Clay Center, Nebraska, those animals were tested for heat tolerance there. And so we took part of that herd, we purchased it, and moved them to Beeville Station. So those animals are a very valuable resource because we know to exact precise percentage the parenthood of the breed type of all of those animals. But in addition to that, each animal has the entire genome mapped. And when a calf is born, we process those animals, we take a blood test, and we send it to them in a partnership. So this was a fantastic contribution that Dr. Milt Thomas made to our station. And so we have equipment to measure weather, and we have measures on the animal, the phenotype, the genotype, additional phenotypes that we're measuring. Oh, excuse me, let me back up, the weather. So this is a comparison of the average humidity temperature index in Hastings, Nebraska, and Beeville, Texas. So I think we're really shocked by this data and how much warmer it is and more humid in Beeville, right? Okay, and so this shows the exact difference mean, and you can see the extra stress going on there. Okay, so we have additional phenotypic measures that we're making on these cattle. We're using a rumen bolus from SmackTech, and that is a lot smaller than my palm. And we bolus it into the animal's rumen, and it stays there. The battery lasts between 5 and 7 years. And we get data like this over time, so every 15 minutes. It shows here the sensor. So we put that in the field, and the animal takes a drink, and then it walks by that, and it takes a reading, and it sends it back up to us. I say us, it goes to Dr. Said. And then already he was able to compare two groups, those with 0 to 15% Brahmin influence and those with 20 to 75%. So we know they're heat sensitive. So this is the temperature difference when you are not Brahmin compared when you're Brahmin. So the other thing we're measuring is GPS colors. And so we know where these guys are going, which has been really helpful for a lot of other reasons too. But so then our wonderful data scientists with expertise in breeding and genetics made this heat map. So this is where our cows were in these different pastures. And so we want to overlay what's going on spatially with our forages to this. So that's where we're headed in the future. So we know where our trees are. We're getting some brush encroachment. Where are they hanging out? So we also have a relative feed intake on these animals. So we're really hoping to make a positive contribution to science. So this is my contact information. I'm happy to answer any questions about forages. So I always liked the second verse better than the first. Strive for ideals high and guard your standards and your traditions. But remember, we hold traditions really strongly here at Tarleton, but it's okay to make it your own. So when I left here, there were no women faculty. There was one female lecturer for equine science. So I really like to see that the faculty are representing the student body a little better. And that's not just here, it's everywhere. But it's okay. And then another thing too is you never know who you're gonna meet. So I'm leaving here this evening to go meet my friend that got me a job with Dr. Muir and spend the weekend with her catching up. I'm so excited to see her. So you really never know So be kind to everybody. And when you climb the ladder, get where you're going, extend your hand out and help everybody else come up behind you. So that's what I think we should all do in our careers. Working with students, even though I'm a teaching appointment, has been one of the most rewarding things that I have done. So thank you all for your attention today. I'm really glad to have been here and been invited. Mic'd already. Any questions?
Thank you. Yes. Thank you for your presentation. I really love your story about what makes you choose your pathway to be a professor. That's really impressive. I have one question. You mentioned about the tillage and non-tillage soil. The non-tillage soil has less like green gas emission. Do you know what's the reason? It's because some physical process?
I didn't present any greenhouse gas emissions data. That was actual soil nitrate nitrogen.
Okay.
It wasn't the emission of nitrous oxide. And so, yeah, so that slide, what is happening probably is we plant the cool season legume probably October in Beeville, October to November, depending on our weather. And there's not really time before we plant, ideally in February, for that plant to nodulate and really contribute nitrogen to the system. And so what's happening is it's taking up nitrogen, it's binding it in the plant material. I think that's what's happening. So we're gonna need to time our nitrogen fertilizer to push the system and get our carbon-nitrogen ratio to work so that there is nitrogen available for the row crop. Does that make sense?
Okay, yeah, I thought
Okay.
maybe there's some microbiology
Yeah.
activity.
Yeah, so we're getting a project started this year with energy cane, which is a hybrid of sugar cane, and some perennial sorghums. And we'll be integrating some cool season legumes into that system. And we have purchased the setup to actually do the greenhouse gas measures for that. So we'll be getting started with some of that work. But yeah.
Thank you for clarifying.
Yeah. Thank you. Any other questions? Oh.
I did have a question about the heat index. Cal, I assume you're measuring growth or weight gain, or will measure that in calves now that you have calves.
Yes.
Is that part of the plan? Because obviously heat tolerance or heat index is one thing, but the rancher is interested in the bottom line.
Yes. So thank you for the compliment, sir. During my time in the lab with Jim, there were 4 of us that ended up getting PhDs during that time. That's just crazy. You're a good professor. His question is about, we have the heat, temperature, humidity index, and are we going to be measuring body weight gain? Yes, there's several measures that we had a first calf crop. Some of the cows that we brought from Nebraska were already pregnant, and then we had a group of heifers, and now those heifers are the ones that are calving now, and the cows will come in just a couple weeks with their babies. But yes, so we process those with a body weight at birth within 24 hours, and then every 56 days until they're weaned. And at that point in time, moving forward, we're going to sell the bulls that are just weaned, because last year we castrated and fed them out. It's just not economical when we're trying to have a cow-calf operation, because we also although this is for research, this is a working ranch, and we sell for sales funds, both our calves and cull animals and hay. So, but yes, we'll have body weights of those, and then the blood sample for the genome mapping. So, and then on the heifers, before they're 18 months old, we'll send them to do the relative feed intake with the Grow Safe system. And that's where we know precisely by bite how much they're taking in and how much they gain. So it's a very encompassing system. So, yes, sir.
If you don't mind. So I find this research fascinating because one of the things that I've seen around the world, it doesn't matter if you're in Tanzania or Botswana or Portugal or Brazil or here, is that ranchers are fascinated with the exterior of their animals, right? The color, the size, the breed. You know, Angus is the best, you know, steak you've ever had. Which is not true, in my case anyway. And so, the approach that you seem to be taking, or the folks in Nebraska took, was, as you described, a motley crew or motley herd. And so, you're looking purely at production and forgetting that whole gobbledygook BS. Trying to be appropriate. About, breed and color and so forth. How much of a cultural shock will your research be to people in Texas, you think, in the future, to be able to tell them, listen, quit producing pure bred Angus or Hereford or whatever, and start looking at actual production indices based on adaptation, not just to heat and humidity, but also KR blue stem, which is here to
Yes.
stay, right?
Yes. So for those of you guys on Zoom, the question is about color and how that is really something that we have preference by cattlemen, and how we're kind of ignoring that in this project with our motley crew of cattle. So I think the cattlemen have a big preference because they're getting dinged when they sell. So our South Texas beef, they get a pretty big ding for too much ear, right, for the Brahman influence, because the idea is that that animal has less desirable marbling characteristics, et cetera, in the meat. So they do face kind of an economic, issue with that. So I think the way this was designed is for breed comparisons. And a good outcome of that could be something like an EPD, the estimated progeny difference, about heat tolerance and grazing ability. So that would be fantastic, right? So is the cow going to actually go out and harvest what she needs and do it efficiently, right? So that's how we can make gains, I think, in our animal production systems. So I do think, yes, there's going to be an issue, but what we might be able to find is one of these mixtures of animal that has really good abilities to thrive in South Texas, but then the ear can be minimized just enough to get under that inspector or the person making the offer, where if you're at the auction barn, right? But yes, it is an issue because people seem to prefer Angus for some reason, but they're not very heat tolerant. So, and then we measured, when these guys were new, something called hair score. It's a 1 to 5 scale. You're shaking your head. You've heard of it before, because that's highly related to heat tolerance. There's some great papers out of A&M Kingsville on that. And the Herefords, they stayed woolly boogers for a long time, and that's not good in Beeville, right? We saw the map of how hot and humid it is. So, and sure enough, that also in their every 56-day weights, they just weren't gaining like some of the other breed types. So, but yeah, it is a good point. There's other ways to use it, though, I guess. So, Any other questions?